I'm Dan. I'm a PhD student in the Dey Lab here at Hutch. This is an anaerobic chamber, or an oxygen-free chamber. All of the bacteria in your microbiome are what's called anaerobes, which means oxygen is very toxic to them. So we have to grow them in an environment that has no oxygen in it, hence our chamber. Now, we get a lot of microbiome samples, either from animal experiments or from people. And from those samples, we can actually culture out the bacteria from those samples. So one thing we do is we measure the growth of those bacteria. We grow bacteria in cultures that are in these little tubes. Now, we measure the growth of bacteria by shining a light through the culture and asking how much light actually gets absorbed. So this is a relatively new culture. You can see right through it. This pen goes right through it. If we have a really dense culture that's been growing for a while, you can't see the pen through it because there's so much bacteria in there. So we're gonna measure how much that bacteria has grown using a plate reader, which kind of shines a light through the bacteria. So I'm gonna load some of this new culture into this plate here, and then some of this older culture that's been growing for a while into the same plate. You can see very clearly there's a big difference between those two. But to kind of quantify that, I'm gonna use this plate reader. So a little arm comes out, I stick the plate on the arm, push it in, and then I go to this computer behind me to take a reading of how many bacteria there are.